The objectives of this assignment are to identify and characterize enzymes that are used in Tide laundry detergent, find gene sequences of laundry detergent enzymes using NCBI, and distinguish genes and genomes using a genome browser. From the Class Smart site, you'll find all the bioinformatics assignments under the Assignments link. Go to the first assignment. The first 3 links are informational websites that you'll use to fill out the homework assignment. The last 2 are your homework assignments themselves. Click on the Excel link, download, and open the table. There are 2 spreadsheets that you'll need to complete. The first table needs to be completed with some of the biochemistry and commercial information about detergent enzymes that are found in cold water Tide detergent. I filled out the first column as an example. I'll take these out now so you can see how I did this. First, go to the Procter & Gamble product guide and find the product ingredients for cold water Tide. Enter these into the row labeled Enzyme Class in Table 1. I'll give you the first one. It's a protease. Novozymes sells enzymes to Procter & Gamble, and their household care catalog is a really good place to find information about their enzyme. The catalog shows the trade names of different enzymes sold by Novozymes, both for laundry detergent and for dishwasher use. Scroll down and learn a bit about detergent enzymes. It's kind of interesting. For each brand, the catalog shows the class of enzyme together with some of the biochemical information. Your task is to find one Novozymes trade brand for each of the 4 enzyme classes in Tide. For example, let's find a Novozymes enzyme that's a protease. It's an enzyme that digests proteins. Look through the catalog to find a protease. And here it is, alkylase. Alkylase is the trade name. Protease is the enzyme name. The easiest way to complete these tables is to use copy and paste. And on a PC, that just is Control-C and Control-V. The biochemical activity is the same as the primary activity. Fill in the cells with the information. The EC number, or enzyme commission number, is the official numeric classification system that describes an enzyme's activity. The EC number has 4 digits separated by periods, each representing a finer classification of the enzyme activity. The EC number is important because it is a standard nomenclature that defines the activity of an enzyme regardless of its name. Complete Table 1 and then move to Table 2. In this table, there are gene ID numbers for 4 genes encoding laundry enzymes. Again, I've given you 1 column, now you need to fill out the other 3. To complete these tables, you'll need to go to the National Center of Biological Information website and do a search on the gene numbers. NCBI has lots of information in many databases, only a few of which we're going to look at in class, so feel free to explore these on your own. Put the gene identifier into the search box and search all databases. You can see that lots of databases were searched. We want the genes database because this is a gene's ID. Now click on the gene. These records are regularly updated and provide the gene symbol, gene description, and locus tag, which you can also use as an alternative to the gene ID if the gene ID doesn't work in some of the databases. Use this information to complete the first 4 rows of the table. Click on the organism's name to find out more information about the source of the gene. Copy and paste the organism's name into the complete cell. Be sure to use the complete name. If the format looks funny, you can right-click on a cell that has the correct format, then click the paintbrush, then drag across the unformatted cells to have them automatically formatted. There's 3 superkingdoms of life: bacteria, eukaryotes, and archaea. These can have very different gene characteristics, so the information can be relevant. For the number of chromosomes and size of the genome, click on the genome link under Entree. We see that 5 Bacillus licheniformis have been sequenced. 3 have been completed and 2 are still underway. 2 of these are identical, and I'll explain that in a second. Find the exact match of your organism and complete the table with number of chromosomes, genome size, and number of genes. The genome size in this case is 4.22 megabases, or 4.22 million bases. Be sure to always use the base units. The reason there are two entries for one organism has to do with how they were bioinformatically characterized. One assembly program found 4,332 genes, the other found 5 less. So don't sweat it, you can use the numbers from either table. Now go back to the gene view. Scroll down to the genome browser. This shows a picture of the genes. The APR gene is shown in green, the red line is the RNA, and the black lines are proteins. Scroll over the gene to get its length and its size. And again, be sure to add the units. In this case, the units are base pairs. The beauty of genome browsers is that you can scroll out to see more chromosomes and a gene's relative position to other genes on the chromosomes. Scroll out so you can see genes on both sides of the APR gene and use this information to complete those two cells in the Excel table. You can scroll out even further to see all the genes, or you can scroll in until you get the genome sequence. Just one more thing. Scroll down to the General Protein Information section. This is the section that gives the name of the protein and, importantly, the EC number. Put the EC number into the table, then go back to Table 1 to collect the information that you've already entered. You can use this directly to complete Table 2. That's it. Save the tables and submit them through the Smart Site assignment section.